Okay. Just a reminder that this coming Monday is our final exam. I only mention this because there is actually a student who didn't know that last Monday was the 4th hour exam. 2:15, we're going to be split between 2 rooms, and the second half of the students will be in Odd 3, which is very near Odd 2, so almost the same, but not quite the same place as before. There are 2 hours allowed for it. The exam will be 50 questions, each worth 2 points. So 100 questions outright, plus I've included 5 bonus questions that are explicitly harder than the others. They're not intended to be easy. And they're 2 points each, so you can get up to 10 points of extra credit that way. Just since people have been asking about the structure of that. Because there was no video from Wednesday, I went back and looked online. And the first session of review work from last year is in the links column if you wanted to look at that from last year. 1 year to the next, biochemistry at this level stays fairly similar. So, any other questions that people have in terms of course matter or course organization right now? Let me log out of this. Okay. Well, let's carry on. Let's see. I think we can go to here. I think we are here. Is this pretty much where we were, folks? Okay. We talked about how beautifully adapted hemoglobin and myoglobin are for transferring oxygen from the lungs to the tissues, because the affinity that hemoglobin has for oxygen is not as great as myoglobin's, and so in the tissues it tends to deliver it to the myoglobin. But that's accentuated by the fact that the presence of protons will decrease the affinity for oxygen. So as the red blood cells get out into the tissues where the protons are there, you've been active, you've got some lactic acid out there, so on, the oxygen comes off, and the hemoglobin can actually pick up the proton to help carry it back out to the lungs. So this circuit helps the body rid itself of the carbon dioxide, excuse me, the carbon dioxide that might be produced during exercise. And this enhanced delivery system, if you like, is called the Bohr effect, the fact that acid contributes to unloading the oxygen from myoglobin. Now, from hemoglobin. Hemoglobin is a tetramer. It has quaternary structure, and it has, in the middle of the complex, in the middle of the tetramer, a hollow space that can actually be seen as a hollow space. And a small molecule, 2,3-bisphosphoglycerate, so it's a bisphosphoglycerate. We're going to see 1,3-bisphosphoglycerate in glycolysis, of course, can fit here in the middle. And when it sits in there, it balances the negative charges there, the positive charges there, and it acts like a little doorstop. So BPG can help stabilize the deoxy form of hemoglobin. And this turns out to be quite important because it changes the affinity curves by making it have a less strong affinity for oxygen. You can see here the 2 curves in the presence and absence of BPG, and so it allows for delivering more of the oxygen in the tissues than if BPG wasn't present. BPG was found here at Iowa and is part of why our blood banking was so significant in the early days. A strategy like this is how the fetus can get oxygen from the mom, because the oxygen affinity of the fetal hemoglobin is a bit higher than the maternal hemoglobin and can get the oxygen away from it if you like. So to shift gears a little bit, we're going to talk about a different constellation of proteins. Another important way that proteins sort of function in the body is they hold cells together. They give cells shape and structural functions. And that group of proteins are called the cytoskeleton. 3 major filament systems comprise that cytoskeleton. The microfilaments are shown here. So we're getting a... You know what? Okay, there we go. The microfilaments are proteins that are abundant, for instance, in muscle cells or in the absorptive little villi in the gut or in platelets, where the platelets are going to contract to make clots hold together. Another group of structures are the microtubules, and microtubules are abundant in long, skinny things like cilia, like neurons. And then the third major group are the intermediate filaments. They're intermediate in thickness between microfilaments and myosin. And these are found abundantly in places where you need to hold stuff together: skin, hair, epithelia, neurons, things that are under mechanical stress of various kinds. This is showing you, again, reminding that microfilaments, which are shown in this picture, were first identified in muscle and then later on in gut. And typically we find that the microfilaments are going to play a big role whenever cells are attaching, adhering to things, adhesion, in contraction, when things get smaller, like platelets, like muscles, or in many cases in stiffness. So parts of the inner ear are made stiff through actin microfilaments. The components of microfilaments, the core of it is made up out of a protein called actin, which is a monomeric protein when it's not in an actin microfilament. And the filaments themselves are about 7 nanometers across, which is why they're small filaments. Here are some microtubules down here. And these are, again, abundant in cilia, neurons, things that are long and skinny. Their stability against in motion allows them to play a role in swimming and in flow. So if you see in the lining of the lung, the cilia that make the fluid move are powered by microtubules. Things that need to be stiff often have microtubules, and they play a major role in mitosis. Oh, Bill. It's alpha-beta heterodimers. So tubulin is a heterodimer in contrast to actin that's a monomer. And they're 25 nanometers across, so they're bigger across than even myosin filaments. Here's a picture of cytokeratins. These are one kind of intermediate filaments. Again, they're abundant in things that have mechanical stress a lot. They give strength against mechanical stress and stuff. They also play a role in attachment along with the microfilaments. And they're more diverse than the microtubules or microfilaments. There are at least 5 major families of them. And so we're not going to explore all the chemistry of those 5 families. They make 10 nanometer filaments, and that makes them intermediate in size between the microfilaments and the myosin. So the names make some sense. And this is showing you how those kinds of chemistries go together. Some of the points here to take away would be that the microtubules have a polarity to them. So the alpha-beta dimer is pointing the same way up the whole microtubule in a helical packing. Here's the actin microfilament. It's a twisted helix of 2 series of monomers, but they're all pointing the same way. The actin has ATP bound to it and is oriented in the same way. Whereas the intermediate filaments are packed without any polarity. They pack together the subunits anti-parallel, and so you end up with like a rope that has no handedness to it. Intermediate filaments are important at the level of structure, partly because they're the first place that we saw a theme that we saw again many different times, and that was the idea of the coiled coil. We talked about the alpha helix being a repeating coil of subunits. If you take 2 alpha helices and they have the right structure to them, can I do this without hitting my poison ivy? You can coil 2 alpha helices together to make a coiled coil, which is obviously likely to be stronger than a single one. And this coiled coil structure is an important part of intermediate filaments, but we see it again in things like myosin and again in things like DNA binding proteins and in a lot of other places. The trick is to have hydrophobic residues at the 4th and 7th position, so when you coil around, the places that touch are hydrophobically in contact. That's the heptad repeat I just mentioned. 2 alpha helices, hydrophobic interactions, and now intermediate filaments can make these coiled coils. They do not have any nucleotide binding. So whereas the actin had ATP bound and the tubulin had GTP bound, intermediate filaments have no nucleotides and are not polar. They have no front or back end to them. And that's just a picture of the heptad repeats. That's showing that at a structural level. Okay? Okay. There we go. Okay. So cytoskeleton, structural proteins. But of course, the thing that we think of proteins most often about, even though by bulk the cytoskeleton is more common, is enzymes. And then certainly enzymes have the greatest variety of structures. So in thinking about enzymes, we're going to embrace the universe of proteins that make things happen. And a lot has been learned by looking at a particular enzyme, something called chymotrypsin, a very simple enzyme. It just breaks stuff down. Here you can see a peptide bond, the peptide bond we talked about on Wednesday. And if you add a water molecule across the peptide bond right there, you recover the carboxylic acid and the amine group that were used to make the peptide bond. Very fast. It's 190 bonds a second, so it's quick at it. 10 to the 11th times faster than in the absence of it. So enzymes are capable of very great accelerations of reactions. Now, if we look at the enzyme, we can see it has a compact structure, and there's a cleft in it. And that cleft is like the center of a hot dog bun, and the peptide backbone slips into there, and that's where the active site is. That's where you're going to cut something. And the two domains kind of pack together to create that active site. It can do this catalysis under mild conditions, so where you might need to use strong acid to break down proteins. Otherwise, here you can do it at not terribly drastic conditions. It has some specificity. We'll probably remember that in a little bit. But thinking about what chymotrypsin does introduces us to the concept of a reaction coordinate, of the course of a reaction, of what happens over time. And if you start off with some reactants, the things you're starting with, you can ask how much free energy is associated with those reactants. The products may be at a different level of free energy. If the products are at a negative free energy relative to the reactants, the reaction will tend to go forward in that direction. So that's the delta G of the reaction. But to get there, usually the reactants have to undergo some kind of conformational shifting around or rearrangement into a transition state of some kind, and that's a barrier to getting over to the other side. If you have a positive delta G for the reaction, it won't be spontaneously in that direction. You might be able to pull it through if you have another reaction linked to it, but it itself would not tend to go forward. But this hill, what's going on with this hill? So if you start with the reactants, in an uncatalyzed reaction, there's some kind of a transition state that has associated with it a delta G for the transition state, or a delta G of activation energy, or energy of activation. They all mean the same things. What the enzyme does is to change things in some way so that the hill isn't so high, that the energy, the delta G of getting to the transition state is not as great. And so more often than in the absence of the enzyme, the energy in the environment will be enough to bump it over the hill and down to the products. It does not change the overall delta G between the reactants and the products. It only changes the height of the hill or how fast the reaction goes. It can't change how far it goes. So it changes the rate of reaction because the height of the hill has been reduced. Any questions about that? Okay. So what enzymes do is they can create chemical conditions to allow for that lower hill, that reduced transition energy, that alternate way through the intermediate path. And they do it a number of ways. Some have cofactors, small molecules that do things that side chains of amino acids can't. There's several common reactions that are recurrent again and again. Acid-base catalysis. There may be a covalent intermediate where the enzyme actually is covalently attached for a while to the substrate. Or metal ions may be playing a role. That basically what the enzyme is a very fancy test tube to hold on to that metal ion to react with the substrate. Chymotrypsin is kind of fun because it uses two different reactions. It uses both acid-base catalysis, and it has a covalent catalytic component as well, where for a little while the substrate is covalently attached to the enzyme. In this particular group of the side chains, aspartate, histidine, serine, this is called the catalytic triad because this is where the actual catalysis happens. This is the active site, the catalytic site within the overall active site. And that triad is preserved in a variety of different compounds. Now, if the catalytic triad is the magic place where the exact changes in the transition state happen, what's the rest of the enzyme for? Well, people have been thinking about that for a while. The first idea was it was maybe like a lock and key, that they fit together so nicely, just like a lock and key. And people began to realize that that created other problems, that if you had a genuine lock and key that was kind of rigid, you might actually make it harder to get over there if your substrates are binding tightly to the enzyme. And so more recent thinking is that there's a combination of different things that come together to make this work. And some of them are very simple, but actually are very powerful. Proximity, holding the reactants close together. You're raising the concentration, in effect. Orientation, you're not just holding them close, but you're holding them pointing a particular way towards each other. Some enzymes undergo induced fit, that they are maybe open and floppy until the substrate binds and then they close on it. There can be an electrostatic component. There can be, in effect, a little slab of positive charge or negative charge that holds onto things to orient things. And another important aspect of it is that in many cases, depending on what the reaction is, the active site will exclude water. Just as the hydrophobic pocket of myoglobin and hemoglobin excludes water from the heme group, it can be very valuable that the enzyme keeps water out so that the only things that can happen are the desired reactions. So looking at these enzymes, these proteases, has actually given us a glimpse of a pattern we see again and again and again in biochemistry, which is of how these things can arise. So if we compare three different proteases in this particular family, chymotrypsin, which we've been talking about, trypsin and elastase, and we compare their tertiary structure, their packing in three-dimensional space is quite similar. This is like the hemes, like beta globin, alpha globin, and myoglobin. And again, like them, there's a lot of divergence in the primary structure, the primary sequence. So there's several different ways to arrive at the exact same tertiary structure. However, all of them conserve that catalytic triad there in the middle. So there are absolute conservation at the reactive sites, considerable conservation at structurally important parts, and then at the surface, out where it's facing the water, much more variation. Serine versus a threonine, glutamine, aspartate, they're all going to interact with water. No real constraint on what that's likely to be. So that's kind of the shape of those proteases. How do we get specificity out of it? And it turns out that as up here at the top is the bond that's going to get cut. If we look at the specificity pocket, it turns out that near the catalytic residues, but not exactly them, near the catalytic residues, the shape of the enzyme holds the side chain in a way that allows the enzyme to be selective. And so chymotrypsin has a large hydrophobic pocket here. Trypsin likes to cut after lysine and arginine, and it has a negative charge at the bottom of the specificity pocket, so that holds lysine and arginine nicely. And elastase tends to cut after smaller hydrophobic things, and it's got these things sticking into the specificity pocket that only let small things fit in. So the specificity pocket depends on shape and so forth and charge to select things. The catalytic residues do the highly precise work on the exact bonds that are going to be rearranged. So that lets us start to think then a little bit about how fast enzymes do it, what we learn about the enzyme looking at that. And if you have a lot of the substrate that the enzyme is going to work on, we're going to start by thinking of a really simple one, of A goes to B. Really simple one. You have lots and lots of substrate. If you add more enzyme, you'll get a faster reaction initially. But suppose you hold the enzyme constant at some fairly small number, and you start increasing the substrate. You can then see that while initially there may be a linear or nearly linear response, at some point you're going to end up going as fast as that particular amount of enzyme can catalyze a reaction. And the shape of this curve should remind you a little bit of myoglobin. This is a hyperbolic curve. We say that it's showing saturation, that it's approaching the point where every molecule of enzyme that can have a substrate bound to it has a substrate bound. We can be slightly more quantitative about this. I'm not going to rederive this, if we look at this hyperbolic curve again, where the velocity initially is rising and then tapers off, we can talk about there being a Vmax, the maximal velocity, no matter how much substrate there is. And this is somewhat more realistic than many graphs because it doesn't actually hit the Vmax, which is true. That when you have half saturated the substrate, the enzyme, sorry, with substrate, that's giving you a measure of affinity. So at the concentration of substrate that produces a half maximal reaction, you have saturated half the molecules with substrate. And this tells you how much substrate has to be around to do that. So this is a measure of how much affinity the enzyme has for its substrate. So the Km is telling you something about affinity, goodness of fit between the enzyme and the substrate. And there are various conditions that make that more precise. So Vmax, how fast the enzyme goes when it's completely saturated. Km, how much substrate it takes to saturate roughly half the molecules. Now, many reactions are much more complicated than A goes to B. And you may remember this one from the pentose phosphate pathway, a fructose 6-phosphate going to xylulose 5-phosphate. And you can see all these different things that are coming and going on transketolase. Every one of those little arrows has rate constants. But you can still talk overall about going from fructose 6-phosphate to xylulose 5-phosphate and talk about a catalytic rate for the thing overall. You don't have to, you can look at the micro constants, but you can also talk about the overall reaction. Now, some enzymes are more complicated than that. Some enzymes don't have Michaelis-Menten kinetics. And in general, when you see a sigmoid curve like this is a sigmoid curve, sort of S-shaped, like a really sloppy S. We saw that in hemoglobin. It was a sign of cooperation then. In general, sigmoid kinetics for an enzyme will generally point to cooperation of some kind or another. It arises through allostery, which means that something binding at one site changes the behavior at another site. Allostery is just Greek for another site. So an allosteric interaction, something binding here changes over there. We can still talk about a Vmax or a Km. There's still going to be some maximal velocity when it's fully saturated. There's still going to be some concentration of substrate that half saturates it. So we can still think about its affinity and how fast it goes when saturated, but measuring it is obviously going to be a little trickier because of the sigmoid shape of the curve. Now, both in practical terms, in terms of biotechnology and stuff like that, and also inside of our bodies, sometimes you don't want an enzyme to do things. Sometimes you want to be able to inhibit it. And sometimes enzyme inhibitors can tell us a great deal about how things work, or they can help keep our cornfields clean. Roundup is an enzyme inhibitor. And enzyme inhibition comes in several flavors. An irreversible enzyme inhibitor is often an analog of the substrate. It looks like the substrate. So here's an example of an irreversible inhibitor for chymotrypsin, and DIPF looks like its normal substrate. The enzyme starts reacting on it and makes the covalent intermediate and now can't let go. And so an irreversible inhibition is an actual interaction between the two that won't ever go away. Often at the active site, it could be off to the side. That is another place that it can happen. And we sometimes call these things suicide substrates. Another common form of inhibition is competitive inhibition. And in competitive inhibition, the substrate might fit into the little pie shape like that. The inhibitor looks like it partly, but not completely. And so they're competing for a common site. And if it's a true competitive inhibition, it will always be reversible. It's just a matter of who's getting there at the particular moment. So it would have to be structurally similar, but it would not be reactive. And when we look at competitive inhibitors, looking at a hyperbolic curve, if the only thing it's doing is competing at the active site, when you add more and more substrate, at some point the odds that the enzyme will see a molecule of substrate and not an inhibitor become essentially 1. And so one way we can tell a competitive inhibitor is that the Vmax, the maximal velocity when fully saturated, is not changed because the outcompete the competitor. But it shows up in the form of a change in the apparent affinity, that it takes more of the substrate to get to the half maximal velocity. Because some of the molecules there aren't substrate, some of them are competitor, and you have to make up for that. Allosteric inhibition is a form of inhibition where you bind somewhere else than the other site and you can change activity. And this gets used a lot during metabolism. So you can have an enzyme like phosphofructokinase in glycolysis. Downstream, quite away from it, is phosphoenolpyruvate. And yet it can come back and bind to the enzyme allosterically and reduce its activity, which is a way to accomplish feedback inhibition. I've made an accumulated PEP, turn down phosphofructokinase. We don't need you right now. But you can also have an allosteric stimulation in contrast to the inhibitors, which, for the most part, the organic chemists haven't found stimulators of enzyme activity. That's built in. And this is just looking at phosphofructokinase in a little more detail. In the absence of phosphoenolpyruvate, there's hyperbolic kinetics. In the presence of phosphoenolpyruvate, we turn those hyperbolic kinetics into sigmoid kinetics because it's binding allosterically somewhere else. But the Vmax is unchanged. It's changed the shape of the curve. It's changed the affinity. It's very much greater, much lower affinity, much higher Km. And what the PEP does is it sort of makes the molecule tense. It's hard for the substrate to bind. ADP would be a lower energy intermediate. It's a signal that the cell does need this reaction, and it can help keep phosphofructokinase in a more relaxed state. Phosphofructokinase is the key to glycolysis, so this kind of allosteric regulation is very important. Another big group of proteins is found in a different kind of an environment, in the membranes. But to think about membranes, we're going to have to think about lipids a little bit. And lipids come in provide for membranes which can make a boundary around cells and so forth. Membranes are both protein, roughly half protein and half lipid. The proteins often have sugar on their outside, so this structure actually has 3 of the 4 major classes of molecules. The bilayer part provides the barrier. The proteins provide the more complicated functions. Membranes are in contrast to things like DNA or hemoglobin in that they are formed non-covalently. They do tend to be asymmetric. The lipids in them are free to move sideways, so they form a fluid mosaic of lipids. The proteins in the membrane tend to be less free to move, and it's been called a solid mosaic for that reason. There's usually an electrical charge across cell membranes. In a living cell of almost any kind, there'll be an electrical charge across the membrane. That electrical charge helps the cell control what's inside and what's outside. And these are just some diagrams of different lipids. Considerable polar head groups on the phosphoglyceride and sphingomyelin. Cholesterol just has this little bitty tiny alcohol group down there. And this one actually is, I've said a fair bit of this so far. This is just a reminder of some of the classes that an integral membrane protein passes through a membrane and has hydrophobic side chains sticking in contact with the hydrophobic part. Peripheral membrane proteins are at the edge, the periphery of it. This one doesn't have a picture for the lipid-linked, lipid-anchored ones. And the kinds of things that membrane proteins can do, they can be channels to let things flow down, they can pump things actively, they can be receptors that can detect hormones or ligands of various kinds, and there are even enzymes on the cell surface that can catalyze reactions outside the cell for things that are useful to the inside or on the inside surface of the cell. And this, I think, I have talked most about, covered most of it in talking about the fluid character of the lipids, the much more solid-like character of the proteins where they move much less. One point is that the lipids in the membrane can move side to side or laterally. It's difficult for them to spontaneously move from one leaflet to the next one because you take a polar head through a lipid interior, but there are enzymes that can catalyze that. But because it's hard to flip them, it means that the outside leaflet can be quite different than the inside leaflet. And just a reminder that this term solid mosaic model isn't in your textbook. So, transport can be passive, where things just go down the gradient, like the acetylcholine receptor, which lets sodium ions in. You can have active ones like the sodium ATPase that can pump sodium out of the cell. Most of the pumps on the cell membrane are powered by ATP. And the cystic fibrosis protein is an important example of a membrane protein that is a pump. It's actually the chloride pump. In addition to gradients that are created by pumps, some of the transporters can use those gradients for what's called secondary transport, where you can couple something to that. Channels can be selective. You can select sodium or potassium and have one specific one. And I mentioned that the gap junctions let cells talk to each other up to things as large as glucose even. So this is just a summary of what I've said, so I'm going to skip it if you don't mind. Okay. Probably the most complicated part of a membrane are membrane receptors. Information outside the cell has to get inside the cell, but you don't want to breach the membrane, and so receptors can pull that trick off. The signal might be a hormone, it might be an amino acid, but there's something or other that can react with that to receive that signal. There's some form of amplification inside the cell, and then that amplified signal is transduced or turned into some kind of response that's appropriate for that event. Protein phosphorylation is one common strategy in getting this to happen, and everything that gets turned on at some point will get turned off. This is showing a diagram of the 7 transmembrane domain receptors as a group of things. They have, this is a blow up here of a G protein, the alpha, beta, gamma G protein, and it's the alpha subunit that can bind GDP or GTP, which is why they're called G proteins. And so when a receptor is activated, it can activate a G protein. That G protein subunit can then go and turn on other things. It can turn on a phospholipase or adenylate cyclase. In the case of adenylate cyclase, then you get cyclic AMP. It's the AMP that then can make other targets be active, such as protein kinase A. This is just sort of pointing that there are parallel pathways in some other cells in which the G protein might target phospholipase C instead, and which in turn produces other secondary regulators. And this also targets a protein kinase, but this protein kinase is protein kinase C because it's calcium activated instead of cyclic AMP activated. And both of these kinases will target serines and threonines on specific targets. And so the common activator up here can have very specific targets at the bottom depending on what's there to be targeted. And some of the things that work this way include glycogen metabolism. There are genes that can get turned on and off through cyclic AMP, synaptic transmission in some neurons. So it's a very flexible system that can accomplish a lot of different goals for sending information around. Another strategy that some receptors have is that when they bind their ligand, they can form a dimer, and then the intracellular parts of it have an activated kinase at that point that's a part of the whole protein, and then that kinase can then have its own catalytic cascade to activate secondary messengers. So we're not going to complete the handout in the remaining 20 minutes, 15 minutes, but I'll see how far we can get into this, and I hope that the rest of the handout will be useful to you. So we'll see what we can do without being too helter-skelter. We've talked about structural biochemistry. Metabolism is the work by which cells can take stuff out of the environment, like energy or reducing power or solid stuff, and then use that to build small molecules like glucose and fatty acids, or the big ones like DNA and RNA. Pathways are often coupled to each other. They get coupled through common intermediates is one common way of doing that. A common strategy is to oxidize carbon fuels, to burn things to get energy. And in many cases, there are motifs that are reused in different parallel pathways. So burning fuels to carbon dioxide and water provides energy. Broadly speaking, catabolism. You can always keep catabolism and anabolism separate. Anabolic steroids build up muscles. Anabolism is building up stuff in general. Leaves you catabolism for catastrophes and breaking stuff down. So catabolism burns stuff. Glycolysis and oxidative phosphorylation are major components of catabolism, but there are others that we've talked about. Catabolism is going to provide usable energy in some small molecules that we can then build up through anabolism. These are some anabolic pathways: gluconeogenesis, meaning making new sugar; glycogen synthesis, making glycogen; photosynthesis. But we had other anabolic pathways too. And a few pathways that can both build up and break down. And so we call them amphibolic. They can both be anabolic and catabolic. The citric acid cycle was the first one we saw like that, that's both anabolic and catabolic. The pentose phosphate shunt is both anabolic and catabolic. And there are some others if you keep thinking about it a bit. Over and over again, we saw compounds with high phosphoryl transfer potential, the delta G associated with transferring or phosphate groups, which is often called a high energy bond. So ATP and GTP have high energy bonds, if you like, in these two bonds out here. Phosphoenolpyruvate, there's another high energy bond there. Creatine phosphate, 1,3-BPG, there's another high energy bond. These are all phosphate bonds that have a high energy of transfer. And we see them in various places, of course. Phosphoenolpyruvate in both glycolysis and gluconeogenesis. 1,3-BPG in those pathways, and in the Calvin cycle, of course. So there are other strategies the cells has for carrying things. So CoA can carry carbon compounds. We saw acetyl-CoA and acyl-CoA and succinyl-CoA. Electrons get carried by various electron carriers. We saw the NAD family, quinones, ferredoxins. Sugars can get carried by UDP and some other things. This is starting to get into an overview of metabolism in general. That the job of glycolysis is going to be taking glucose and other sugars from a 6-carbon down to a 3-carbon fragment. This portion of it can be anaerobic and can be fermentation like a yeast does, or we do when we're doing intense energy. If there's oxygen available, it can be fed into the citric acid cycle as acetyl-CoA. And the reactions of glycolysis have some overlap with gluconeogenesis. There's a lot of overlap with gluconeogenesis. Some overlap with the Calvin cycle. Some overlap with the pentose phosphate pathway. Here we have the citric acid cycle. It's amphibolic in that it takes in these 2-carbon acetyl-CoA fragments, spits out 2 molecules of carbon dioxide. That's the catabolic aspect of it. But it can also be used to provide carbon backbones for amino acids and other things. That's the anabolic side. But the outcome of the citric acid cycle is to take the electrons that came in on acetyl-CoA and feed those electrons into the oxidative phosphorylation pathway of the inner mitochondrial membrane. The movement of those electrons allows for proton pumping, and the proton gradient then can get used to make ATP. And the next several slides are going to be comparisons of some of these pathways in a slightly different way than we did at the original time, as a way for you to kind of see them synoptically, if you like. So here's a pair of pathways: glycolysis, meaning breaking down sugar; gluconeogenesis, meaning making new sugar. And these are similar in that they're both going to use 3 and 6 carbon phosphosugars. They're both of them controlled at various levels. Both of them occur in the cytoplasm of the cells. By contrast, it's glycolysis that's the catabolic one, gluconeogenesis anabolic. Everybody's got to be able to do glycolysis. Gluconeogenesis is pretty much limited to the liver and a little bit in the kidney. Glycolysis, being catabolic, is going to produce ATP and some acetyl-CoA. Gluconeogenesis, being anabolic, uses up both ATP and GTP. Glycolysis is turned on by fructose 2,6 bisphosphate. Fructose 2,6-bisphosphate is this teensy amount of activator made by a different phosphofructokinase that can allow regulation of the pathways. And it will turn on glycolysis, but it will turn down gluconeogenesis. So fructose 2,6-bisphosphate is a regulatory molecule. It's not being used for energy. AMP has, again, opposite effects on the two pathways, as you'd expect. AMP is a low-energy compound. It stimulates glycolysis. It inhibits gluconeogenesis. And they're, at the beginning of gluconeogenesis, the bypass reactions you need to get pyruvate up to phosphoenolpyruvate consume ATP and GTP. A couple of cyclic pathways, the citric acid cycle and the Calvin cycle. These are both organelle pathways. They both have allosteric control for their rates at which they go. They are in different organelles. Citric acid is in mitochondria, so all eukaryotes basically have it. Calvin cycle is only in the chloroplasts, only plants have it. Citric acid cycle is an amphibolic pathway, both catabolic and anabolic. All tissues have the citric acid cycle, whereas the Calvin cycle is really just for building things up, and it's just in the leaves. Citric acid cycle's purpose is, if you like, to release NADH and carbon dioxide. It's burning the carbon to carbon dioxide, and the electrons come out as NADH. And Calvin cycle is going to use NADPH. Remember, the P there is for the anabolic processes for the most part, and it's going to need carbon dioxide. It's the plants that suck up the carbon dioxide. Be nice to your trees. They're your friends. In the case of the citric acid cycle, the compounds that come out of it get used for lots of different things. You can make fatty acids from this cycle. You can make amino acids from this cycle. You can make sugars from this cycle. So if you can get things out of it for lots of different purposes. By contrast, the Calvin cycle has some similarities to actually gluconeogenesis, which is not surprising. We're trying to make sugar with it. So, if we think about the energetics of organelles, we can compare the two big organelles that are so characteristic of eukaryotes in contrast to prokaryotes, the chloroplasts and the mitochondria. Both of them are involved in electron transport. They have electron transport chains in their membranes, and as electrons are transported, they move protons. So both of them create a proton gradient. They have electron carriers. They have quinones that are lipid soluble, and they have water soluble ones like cytochromes and plasticyanin. There are integral membrane protein complexes that do the electron transfer and the proton pumping, so that they have to span the membrane to be able to do that. That proton gradient is how the ATP synthesis is made as long as the membranes are intact. And that ATP synthase in both of them is kind of like a turbine, whereas the turbine, I should say, turbine, not turbine, sorry, kind of a different thing. So it's like a turbine, whereas the protons are flowing around the C ring and out, the ATP is made on the other side. The contrasts then are where the electrons come from. Chloroplasts can strip electrons from water, so they can produce oxygen, and chloroplasts are our only source of oxygen, and blue-green algae. They can consume carbon dioxide. By contrast, the mitochondria are going to use that oxygen as their final electron acceptor and release carbon dioxide. And again, the differences in the tissues. Glycogen synthesis and degradation. So large polymer of glucose, not a very structurally interesting one, just lots and lots of glucoses in kind of a helical spiral that branches a bit. The two processes are going to be concentrated in a few tissues. So glycogen is in the liver and the kidney and muscle. So unlike gluconeogenesis, muscle can take part in this one. These pathways are involved in maintaining a good regular glucose level, and they're regulated by hormones and by local metabolites. But they respond quite differently to different hormonal signals. So epinephrine or adrenaline, which is released in exercise, will stimulate glycogen breakdown, which makes sense. You need the glucose right now. Whereas it will inhibit glycogen synthesis. So you don't have a futile cycle trying to do the opposite things. Insulin has the opposite effect. There's lots of sugar around in the bloodstream. Insulin will rise. You'll make glycogen makes good sense, whereas insulin will inhibit its breakdown because you've got lots of sugar in the bloodstream already. Use different enzymes, so glycogen synthase, glycogen branching enzyme to move the branches around, whereas you obviously need some other things to make glycogen degradation. Glycogen phosphorylase is that very cute enzyme that can take an inorganic phosphate and break a bond by adding a phosphate to it so that the thing that comes off is a glucose 1-phosphate. It doesn't hydrolyze the bond, it phosphorylizes it. It splits it with a phosphate. Oops, sorry. Let's go back up again. Amino acid synthesis and degradation. You didn't know you could summarize these on one page, did you? They often involve transamination reactions. So you can transfer a nitrogen between two compounds if there's a carbonyl oxygen across from it. So the transamination reactions are free reactions that allow moving that nitrogen around a lot. A lot of these reactions are connected to the citric acid cycle. But they sort of differ in the nature of that reaction. Amino acid synthesis will consume citric acid intermediates, whereas when you have an excess of protein and you're breaking amino acids down, you can feed them into the citric acid cycle. It costs energy to make amino acids. If you have an excess of protein, you can burn amino acids and get energy out of them. Amino acid synthesis principally happens in the fed state, whereas amino acid degradation will happen in the fasting state or when you've had a great deal of protein over and beyond what you need for the immediate needs. Fatty acid synthesis and beta oxidation. Oh, my beta stayed. Well, that's interesting. Okay. I don't understand. There's a lot I don't understand. So fatty acids, of course, are these long-chain hydrocarbons that have a carboxylic acid on one end. Membranes, energy storage, stuff like that. Both of these processes are going to use CoA cofactors, and the chain length will change by 2 carbons at a time. When you're making fatty acids, you're going to use up citric acid intermediates, such as citrate. It's going to cost you energy and NADPH because it's a synthetic process. It'll be principally happening in the fed state, and it'll happen if you have an excess of fat or if you have an excess of sugar or an excess of protein. You can make fat out of anything, unfortunately. By contrast, beta oxidation is going to take off in 2 carbon fragments, produce acetyl-CoA fragments that can be then burnt in the citric acid cycle. It produces NADH. The NADH will be fed into the electron transport chain to produce more energy. NADPH was being used to synthesize. NADH is used for energy production. As you're doing beta oxidation, ultimately it'll come out as carbon dioxide. And again, beta oxidation is principally going to happen in the fasting state, not in the fed state. Pentose phosphate pathway, or pentose phosphate shunt, sometimes called. This is kind of a nifty pathway for doing a lot of different things, and we're not going to try to pin them all down in review, but sort of hit the very highlights. Glucose 6-phosphate could go to glycolysis, of course, but it can also go to the pentose phosphate pathway. And in the pentose phosphate pathway, you can burn glucose in the cytoplasm. Carbon dioxide comes off as a carbon dioxide. And in the process, it produces 2 molecules of NADPH, good for synthetic processes. That leaves you with a 5-carbon molecule that could be used as ribose. Ribose is good. Or could be fed in here and start looking. Some of these things look like citric acid intermediates, right? Here's glyceraldehyde 3-phosphate. So you can feed that 5-carbon backbone through reactions that end up producing citric acid cycle intermediates in a non-oxidative way. So you have an oxidative phase that produces NADPH and a non-oxidative phase that can produce citric acid intermediates. It can be interesting to think a little bit about the evolution of metabolism, that it hasn't always been exactly the same way all the time. Glycolysis almost appears to be 2 pathways, that the top half of the pathway is all hexoses and the bottom half of the pathway is all trioses. And interestingly, it's the triose part of the pathway that's most conserved. Across the widest variety of organisms, the bottom half of the pathway is the most the same. We don't really know whether that bottom half was first made for energy production or for biosynthesis. Another place where we can make suggestions about evolution is in the citric acid cycle. Blue-green algae don't have it because they miss one enzyme, alpha-ketoglutarate dehydrogenase. So perhaps this was initially a biosynthetic pathway for blue-greens, and that by inventing alpha-ketoglutarate dehydrogenase from its very similar companion, pyruvate dehydrogenase, suddenly you have the power of the citric acid cycle and the potential for aerobic life. Photosynthesis, the earliest photosystem we know is like photosystem 1, and it can use hydrogen or other things as their electron donors, but it doesn't make oxygen. It took the invention of photosystem 2, blue-green algae, old as they are, still were able to come up with photosystem 2 3 billion years ago, make oxygen for the atmosphere, make life possible. And that's 20 after, and how can I mind stopping on making life possible? Thanks very much. I hope you've enjoyed it and learned biochemistry. Okay, cut.